Hello everyone, this is Anisa Rizvi, MSc in Clinical Microbiology, and today we are going to discuss about the world of microorganisms. The branches of microbiology can be classified into applied sciences or divided according to taxonomy as is in the case with bacteriology, mycology, protozoology, virology, and phycology. There is considerable overlap between the specific branches of microbiology with each other and with other disciplines, and certain aspects of these branches can extend beyond the traditional scope of microbiology. A pure research branch of microbiology is termed as cellular microbiology. Microbiology is the study of microorganisms, those being unicellular that can be single cell or multicellular that can be cell colony or a cellular that is lacking cells. Microbiology encompasses numerous subdisciplines, including again virology, bacteriology, mycology, it can be immunology, and parasitology. Then we are going to see what is eukaryotic and prokaryotic organisms. Eukaryotic organisms possess membrane-bound organelles and include fungi and protists, whereas prokaryotic organisms, all of which are microorganisms that are conventionally classified as lacking membrane-bound organelles, and it can be included as bacteria and viruses. Microbiologists traditionally relied on culture, staining, and microscopy. Now let us see what is there inside of this world of microorganisms. As you can see in front of you, there is an instrument called microscope. This microscope is used to observe microorganisms. Then the question arises that what is microorganism? Microorganisms are unicellular or multicellular organisms that are microscopic. The study of the microorganisms is called microbiology. There are microorganisms, bacteria, and planktons. Different type of microorganisms are seen around us. Now, where are they present? Question in front of you that where we can see these type of microorganisms. As you know that these organisms are invisible to us, we are not able to observe, we are not able to physically see these type of organisms. So the question arises that where are these type of organisms present? They are found everywhere, in water, in soil, in air, desert soil like them, because of hot and dry climate, but found nearly roots of plants and dead leaves. Everywhere you will find microorganisms. They have heterotrophic mode of nutrition. As you know, what is the meaning of heterotrophic mode of nutrition? There are two different types of mode of nutrition, that is autotrophic and heterotrophic mode of nutrition. So all the bacteria, all the viruses, all the protozoa, and all type of microorganisms have heterotrophic mode of nutrition. That means they are depend on others for their food. They cannot prepare their own food. Parasitic microorganisms causing disease in hosts, and they are called as pathogens. Now we are going to see what is the meaning of bacteria. The most abundant organism on earth. It lives everywhere in soil, water, plants, and animals. Bacteria vary in their shapes, like bacteria have spherical, it can be bacillus like rod shape, it can be spiral, that spiralum shape, and it can be arranged in different ways. Okay, besides this, they are organized in specific cellular shape. Bacteria reproduce asexually and divide rapidly. Bacteria damage human tissues by producing toxins and cause disease, that is food poisoning. Now we are going to see what are the viruses. The viruses also, as you can see, that they are also invisible creature in front of us. So they are called as microorganisms, which we cannot see with our naked eyes. So we have to use microscope. Different type of disease are caused by viruses, and viruses are also called as pathogens because they cause disease in human beings. Defense against viruses. The first line of defense against the viruses, that skin and mucous membrane, which also line the gastrointestinal and respiratory passageways. Skin is tough and stomach acidity act as a disinfectant. As you know that stomach is producing different type of we can say the acid which is uh, protecting even our stomach, the lining of the stomach, and that acid is called as hydrochloric acid. This hydrochloric acid is not only provide the acidic media for the food, but it is also act as a disinfectant for the pathogens, for the microorganisms which are entered along with our food. Second line, after the viruses enter the blood and other tissues, white blood cells and related cells, that is phagocytes, consume them. Accumulation of phagocytes in area of infection is known as pus. As you can see, the pus in case of some infection, skin infection, ear infection, nose infection, body, any part of your body, if they are having some swelling or some pus-like structure, some draining is coming out from that particular area, that is called as pus. And pus is nothing but accumulation of phagocytes in area of infection. We are going to see what is the meaning of phagocytes in the next step. So now, what is the meaning of phagocytosis? As you can see in the first picture or first diagram, that it is innate non-specific immunity. Okay, these are the phagocytes or phagocytosis process. You can see in this cell, this is a white cell. Okay, as this pathogen enter inside our body and they are having on their bodies proteins, the spike of proteins, pathogens are carrying, pathogens are nothing but microorganisms. It can be bacteria, it can be viruses and anything. So this pathogens enter inside our body. And now what will happen the next is this is the phagocyte. Okay, this phagocyte is having receptor on their bodies. In the center is the nucleus and outside of this body they are having some special type of structure that is called as receptors. These receptors are taking these proteins inside them. Okay, when they are taking these proteins engulf, or we can say they are taking inside, the pathogens interact with phagocyte receptors. Okay, these are the pathogens and they are interacts with the phagocyte receptors. You can see, and then what will happen that phagocyte pathogens envelop. Okay, now what will happen that they will open their this area and they will engulf all this pathogen inside. Okay, inside this phagocyte, there is an enzyme that is called as lysozyme. These enzymes start digesting. Okay, in this case, you can see the digestion of the pathogen is going on with the help of this enzyme that is lysozyme. And then what will happen the next, then major histocompatibility complex will form inside the body of this phagocyte. The pathogens break down into proteins and other molecules. Then lastly, you will see that antigen presenting cells, it will be going outside the body. Means they will be taking out in the form of adaptive immunity. Okay. So phagocyte is the process by which a cell uses its plasma membrane to engulf a large particles or a large particle, giving rise to an internal compartment that is called as the phagosome. It is one type of endocytosis. In a multicellular organism's immunity system, phagocytosis is a major mechanism used to remove pathogens and cell debris. As you can see, that when the first will form, so these are the phagocytosis process, and this is the phagocytos or phagocytes, you can say, that will be engulf all the debris which is forming around that particular pus. So as a result, after some time, we are going to see that all these debris will be vanished because of this phagocytes and the process of phagocytosis. As you can see, this phagocytosis, they are a key component of the innate immunity system. We are having different type of immune system, and that one immune system is called as innate immune system. There are three main groups of phagocytes: monocytes and macrophages, granulocytes and dendritic cells, all of which have a slightly different functions in the body. Several types of cells of the immune system perform phagocytosis, such as neutrophils, macrophages, and dendritic cells, and B, lymphocytes. Another function of phagocytosis in the immune system is to destroy pathogens like viruses and bacteria, ingest and infected cells. Phagocytes type of cells that has the ability to ingest and sometimes digest foreign particles such as bacteria, carbon, dust particles, and dyes. In the blood, two types of white blood cells, neutrophilic leukocytes, that is microphages, and monocyte, that is macrophages, are phagocytic. Means they are performing phagocytosis. Okay, so as a result, you can see if in our body some foreign particles like bacteria, like carbon, like dust, and dyes, if anything will be enter inside the blood, so these are the really they are performing their role. These phagocytes, these type of cells will engulf these all foreign particles, and they will be destroyed these all foreign particles. Okay, now again we are going to see what is this phagosome, that like if you can see here that bacterium is there, that is there are some special type of receptors are present on the bodies of the phagocytes, and they are attracted towards, means there the protein which is on the body of this bacterium will be combined with the receptors, and this bacteria will be enter inside the body. As they will be enter inside the body of this phagocyte, that is during the process of phagocytosis, these phagosomes will be moving towards phagolysosome with the help of lysosome enzyme. Okay, and then as a result, everything will be breaking down, and the soluble debris will be going outside the cell, while the insoluble will be staying inside the cells. Okay, this is called as exocytosis. Now, humoral immunity. Some different types of immunity you can see that in which you can see here. This is antibodies which are having receptors, and these are antigens. They are having special proteins. They both will be combined together, and as a result, they will be working against that particular foreign body or bacteria or viruses. Now we are going to see about the viruses. Some microscopic entity consisting of a single nucleic acid surrounded by a protein coat and capable of replication only within the living cells of bacteria, animals, or plants. Viruses particles are unable to make copies of themselves. They must infect living hosts to make multiple copies. Viruses that infect bacteria is called as bacteriophage. Now, viruses cause disease in animals as rabies and different foot mouth diseases. They cause serious diseases in human beings and like HIV or hepatitis or common cold or influenza and polio, etc. Then this is the structure of different type of viruses. You can see there is a tail and tail fibers are coming and this is called as capsid head and this is a tail bacteria that by different type you can see the different shapes of bacteria in that you can see tailed viruses and filamentous viruses. Filamentous viruses different in the structure but the main thing is their nucleic acid both are having nucleic acid inside their cells. Okay, that is capsid filament. It is here you can see the capsid head, but both are carrying this nucleic acid, which is the most important structure inside the virus's body. Viruses survive in soil or groundwater. Non-enveloped viruses, groundwater velocity models, like they cannot, if they are having envelope around them, they can survive for 7 days. But in case of non-enveloped, they are not able to survive for more than 7 days. Okay, now soil viruses, there are ultra microscopic algae uh, there are ultra microscopic obligate parasites. They have head tail structure with diameters 0.05 to 0.1 microns. Always require a living host for their multiplication. As you can see, there are these are the viruses which are yellow in color and the plant pathogens which are red in color. They are much smaller than bacteria, only visible through electron microscope. So not only they are harming animals and plants, but animals and human beings, but equally they can be harmful for the plant's body. These are different type of bacteria in front of you. You can see the different type of structure of like this is the Salmonella typhi which cause typhoid in human beings. If you can see it is a cholera, Vibrio cholera that will be causing or if they are the positive agent of for cholera and for the tetanus, Clostridium tetani is the positive agent for the tetanus and this is for the pneumonia. This type of structures available. This is Staphylococcus aureus and different type of bacteria in front of you. You can see this is a typical structure of bacteria. If we are going to see the structure, we have to move from outside to inside. So we are going to start from the flagellum, which are responsible for the mortality, or we can say that movement of a bacteria. This is the structure of a bacteria. Okay, this is the flagellum. Then we are going to see what is there outside. Okay, So the next part is called as the capsule. Outside blue color structure, the blue color you can see this is the capsule. Okay, flagella will be attached from the inside, but outside first structure is called as capsule. Okay, all the bacteria usually uh, encapsule, or you can see that they are covered with a structure called capsule. After this capsule, we are going to see the next part, that is cell wall. You can see the cell wall is covering all over the body. Then the third structure, that is called as plasma membrane. So that is the reason that bacteria are very much resistant. Why they are resistant? Because they are engulfed, they are encapsulated in a structure called capsule. Then the second structure is the cell wall, and the third structure is the plasma membrane. So it is very difficult for the antibiotics to uh, remove this capsule, then the cell wall, then cell plasma membrane. Then only we can kill that particular bacteria. Okay. Now the next part which we are going to see, it is a small structure pillars that is called as. Then inside, when we are enter inside the cell, we are going to see this is cytoplasm. All over the body there is a cytoplasm, and inside the cytoplasm we can see the small structure that is called as ribosomes. Okay, now beside this ribosome there are small structure that is called as plasmids, and in the center you can see there is, without any covering, without any nuclear membrane, there is a structure that is called as nucleotide. Nucleoid, that is DNA, deoxyribonucleic acid. This is all about the structure of a typical bacteria which we are going to see. As you have seen that flagellum is present, cytoplasm is present inside, and that the outermost covering is called as capsule. The second covering is called as cell wall. Then third covering is called as plasma membrane. And plasma membrane is covered. They are protecting this liquid structure that is called as cytoplasm. Cytoplasm carries so many bodies in which ribosomes and plasmids are present, and in the center, without any covering, nuclear covering, there is a structure called DNA, and that is called as nucleoids, okay, which is present in bacterial cell. Now, bacteria are microscopic, single-celled organisms that exist in their millions in every environment, both inside and outside other organisms. Some bacteria are harmful, but most serve a useful purpose. They support many forms of life, both plant and animal, and they are used in industrial and medicinal purposes. Bacteria are the plural of bacteria, which are microscopic one-celled organisms. They are found everywhere and can be harmful as an infection, or they can be beneficial as a fermentation or decomposition. Five types of bacteria are, it can be coccus, it can be bacillus, it can be spirulum, it can be bacteria, and it can be mycoplasma. Actually, the most bacteria are good. Comparatively speaking, there are only a handful of bad bacteria out of them. Of the total bacteria we are going to see in our bodies, a healthy balance is 85% good bacteria and 15% bad bacteria. Remarkably, scientists estimate that only 1 to 10% of total bacteria have even been discovered. How about the protozoa? We are going to see what is protozoa. If you can see, this is the structure of protozoa, which you can see in front of you. Protozoa, also protozoan as a plural we can use. It is an informal term for a group of single-celled eukaryotes, either free-living or parasitic, which feed on organic matter such as other microorganisms or organic tissues, or it can be depend on debris. One type of protozoa is amoeba. It is very much familiar to everyone. Amoeba are eukaryotes. Those bodies most often consist of a single cell. The cells of amoeba, like those of other eukaryotes, possess certain characteristics features. Their cytoplasm and cellular contents are enclosed within a cell membrane. Their DNA is packaged into a central cellular compartment called the nucleus. As you can see in front of you, the structure of an amoeba. Okay, if we are going to see from the outside, you can see from outside there is outside, if you will see the finger-like projections, we are going to see that is called as pseudopods. Okay, pseudopods are the false fingers of that particular animal or particular organism. Means the meaning of pseudopodia or pseudopods means false finger. Okay, then outside we are going to see the structure that is called as plasma membrane. Outside covering that is called as plasma membrane. And after this plasma membrane, you can see the dot-like structure. This dot-like structure is called as ectoplasm. Okay. Then in the center, whatever the liquid part is there, that is called as endoplasm. Both are collectively called as cytoplasm. Now we are going to see the other part, that is some crystals are present in this endoplasm. Then you are going to see some structures that is called as food vacuoles, and in the center a round shaped structure is there that is called as contractile vacuoles. In the center, as we can see, the envelope structure is present that is called as nucleus. So apart from this bacteria and viruses, usually protozoas are having enveloped nucleus. Thank you so much, every one of you, and we are going to see in your next video. Hope you will like, hope you understand each and everything which we have discussed today.